All right, so we're going to go ahead and introduce our photosynthesis lab. Today is Thursday, 11/12/20, and as you remember, the photosynthesis equation is shown right here, but it's not balanced yet because obviously if you need 6 carbons for your glucose molecule, you're going to need to add 6 in front of the carbon dioxide. You're also going to need to add a 6 in front of the H2O because you're going to end up needing 12 hydrogens. Then there's going to be 6 oxygens inside the glucose plus 6 molecules of oxygen, which is actually dioxide, right? There are 2 oxygens, so that's 12 total. And that's going to be coming from the 6 here and the 12 here. So remember, equations always need to be balanced. If you already took chemistry, you already know that, and you already kind of know how to balance them. But in photosynthesis and cellular respiration, it's pretty easy because it's almost always a 6. So again, we're going to be going over the photosynthesis lab. I'll introduce it first, and then we're going to collect data. Then after you finish all this, you're going to be writing a report. Yes, you're writing a report this time. There are a couple paragraphs that you do have to write, and I will show them to you on the last page. And then afterwards, please do your Chapter 7 summary and Learn Smart. That is due on Sunday, just similar to how Chapter 6 was due last week, Sunday. The learning objective is still that y'all will be able to construct explanations and representations, and then pose scientific questions about mechanisms, structural features of cells that allow organisms to capture, store, or use free energy. So photosynthesis is obviously one of those processes where you're going to be talking about capturing and storing energy. Okay, so you all kind of know a little bit about photosynthesis already from your regular biology class and even from middle school science. So in this lab, we're going to go through the background. And you should have already gone through this background for your pre-lab quiz that you took on Tuesday. So here's the background. Photosynthesis fuels ecosystems and replenishes the Earth's atmosphere with oxygen. Like all enzyme-driven reactions, the rate of photosynthesis can be measured by either the disappearance of substrate or the accumulation of product or byproducts. The general summary equation for photosynthesis is 2 H2O2 waters plus 1 carbon dioxide plus light energy makes a carbohydrate plus oxygen plus water. Now the carbohydrate we're actually focusing on is going to be glucose. So technically this the numbers shown at the front of these equations are not exactly the correct ones we're going to be focusing on. Remember, we just talked about them. The number we're actually going to see is the 6. Okay. So because the spongy mesofuel layer of leaves is normally infused with gases of oxygen and carbon dioxide, leaves or discs cut from leaves normally float in water. If you throw a leaf in the water, it's going to float. Even if you cut a piece of the leaf, it'll still float because there's gases embedded inside of the middle tissue layer of leaves. What would you predict about the density of the leaf disc if the gases are drawn from the spongy mesofuel layer by using a vacuum and replaced with water? Right, so now if you add water instead of gas, after you remove the gas, the leaf itself already has some density, the material of like the cellulose, leaf cells, right? If you remove the gas that is keeping them floating, what's going to happen to the leaf discs now? Are they going to still float? The answer is no, right? They're not going to be floating anymore, so they're actually going to be starting from the bottom, and that's closely related how we're going to start out this lab. So this is a plant that is underwater, and it's one that you'll find in fish tanks quite often. And I'm kind of curious on what you think these bubbles are made of, right? We know that leaves are doing photosynthesis, so they are producing a gas called oxygen. So some of these bubbles are oxygen, right? But we also know that leaves are also performing cellular respiration since they are living organisms. And if they are performing cellular respiration, they're also producing carbon dioxide and using up oxygen. So these bubbles are not anything like pure oxygen or pure carbon dioxide. But when you actually look at the composition of these bubbles, the percentage of oxygen is higher than the percentage of carbon dioxide in the bubbles, which is pretty cool. So what would you predict about the density of a leaf disc if the gas are drawn? We already talked about this, right? So if you draw the gases out, the leaves are originally floated. If you draw the gases out and replace it with water, then the leaf is going to be equally or even more dense than the water it's surrounded by. And that's because the leaf itself, like I said, the cells already have mass, and that mass is more dense than water. So it's going to cause them to sink. These leaf pieces or these leaf discs are going to sink if you replace the gas with water. So if the leaf disc is placed in a solution with an alternate source of carbon dioxide in the form of bicarbonate ions, then photosynthesis can occur in a sunken leaf disc. As photosynthesis proceeds, oxygen accumulates in the air spaces of the spongy mesofuel, and the leaf disc will once again become buoyant or float and rise in a column of water. Therefore, the rate of photosynthesis can be indirectly measured by the rate of rise of the leaf disc. Okay, that's kind of what we're going to be measuring. However, there's more going on in the leaf than that. We must also remember that cellular respiration is taking place at the same time as photosynthesis in plant leaves. Remember that plant cells have mitochondria too. What else could be going on that might affect this process? Right? Aerobic respiration will consume oxygen that has accumulated in the spongy mesofuel. Consequently, the two processes counter each other with respect to the accumulation of oxygen in the air spaces of the spongy mesofuel. So now you have a more robust measurement tool. The buoyancy of the leaf is actually an indirect measure of the net rate of photosynthesis occurring in the leaf tissue. You can actually see that the rate at which it floats is the rate of the oxygen or the extra oxygen that the plants are producing, right? Because it's using up some of it in cellular respiration and releasing that as bubbles, but the oxygen is being stored inside of its leaves in the mesofuel. So the flotation of the leaf is dependent on how much extra oxygen it is producing that it's not currently using up. And as an example, that would be the oxygen that we have the ability to then breathe because it's extra from what the plants are making and using. So again, this is another picture of the diagram. Right, the mesofuel is right here. So you see there's a lot of space in between the cells of the leaves here because it's spongy and you can press it and compact it and it will spring back because there are gases that are being stored in between, and that gas is oxygen. So in this part of the lab, you will learn how the floating leaf disc technique can measure the rate of photosynthesis by testing a variable that you know affects photosynthesis. Later, you will apply this technique or we'll use a computer-based. We're not going to actually do a computer program. We're just going to be doing another version of the lab to test a variable that we choose. So it is important for you to develop a few skills during this part of the investigation in order to carry out your own investigation. For the floating leaf disc technique, the most challenging skill is to get the disc to sink. Don't just watch somebody do this, make sure you can get the disc to sink as well. And that's kind of ironic because you're going to be watching a demo and you're just going to be watching me do it. Okay. So I'm going to pose a question on how you're going to write your hypothesis right now, but you are going to write it later on. Okay, so just have it in your mind. So I'm going to be asking you to write a hypothesis about which cup will float faster. So one of our cups is going to have just water in it for the leaves to try to, do photosynthesis and float. And the other one is going to have water with sodium bicarbonate, which is going to produce the bicarbonate ions, which are an alternate carbon source, right? Obviously, if they're underwater, they're not going to be exposed to the air where carbon dioxide is. So they're going to be getting this alternate source of carbon coming from baking soda, which is sodium bicarbonate. Okay, and I wrote my labels on my cups as baking soda, and I shortened it to BS, so we're going to just be referring it to BS. Okay, so you're going to be writing this as an if-then statement. You're going to be comparing the cups, right? So you're going to be saying something along the lines of, if the leaf discs have a carbon source in the sodium bicarbonate cup, then they will rise at a faster rate or a slower rate than the cup without a carbon source. And so you're going to be telling me, do you think they're going to rise faster or slower? And it's a pretty straightforward one because everything else about them is going to be the same and kept constant. It's just the only thing different is that whether or not they have a carbon source. So if you look at the diagram, this is basically what we're going to be setting up. I'm going to be providing light to both of the cups and both of the environments, but we're going to be starting off with removing the gas within the leaf disc. And that's going to be done with a syringe. I'm going to be putting in the solution that belongs in either cups with 10 or so leaf discs, and then holding the top of the leaf disc to then pull the syringe down and vacuum out all of the gas inside of the leaves. Now, I'll be able to tell if I succeeded or not based on whether or not the leaf discs all fall to the bottom of the syringe. And if I didn't, I can try 1 or 2 more times. But if I keep trying, it's actually going to bruise the leaves and actually destroy the capillary internal systems of them. So then I would be forced to go and puncture more and then start all over. So the idea is to try to get it on the first try. So you pull down the syringe and then hold it for about 10 seconds, and I will demo that shortly. Afterwards, I will be putting them into the cups with their respective solutions, one with baking soda and one without baking soda. And note that there is a drop of dilute liquid soap added to each of the cups because without the soap, you actually can't fully extract the gases from within the leaves. And it's just something that has to do with the stoma being open and the leaves having interactions with the cells inside of them. So they're going to hold on to gases because that's part for their survival. So the soap actually helps remove all the gases since these leaves are going to be dead anyways. I have already punctured the holes out of the spinach leaves. We're using spinach. We're not using kale or anything else. And I've punctured over 20, so we're going to be having more than 10 in each one, but we'll only be recording until 10 leaf discs rise up from the bottom. So you don't have to worry about them being too many inside of a syringe or inside of a cup, because we're just going to be taking into consideration when 10 are done doing their job. And these are all the steps that you should have already understood and wrote down and paraphrased for yourself inside of your pre-lab quiz. And I'm not going to go over them because you should have already read them. Okay, and then here's a note that I'm going to be saying that I'm not going to be using natural light just because in my room there's no good natural light since the sun doesn't really hit the windows here at all during the whole day. So I'm going to be using a fluorescent lamp, which is the same thing that people use when they grow plants indoors or in the dark, so that the plants get enough radiation to get light energy and store that light energy. So we're going to be checking those every single minute. And on the minute, you're going to be counting how many are floating and how many are sunken. And it's an easy kind of calculation to figure out just because we're going to be wanting to just count how many are floating as the focus. I don't actually think you have to count how many are sunken. 1, 2, 3, 4, 5, 6, 7. I will count them for you though. Okay. And you're going to be recording data on a table similar to this. You're going to want to leave some space at the bottom because we don't know exactly how long these are going to go for. Based on past experience that I've done in the past couple of years, they can end as fast as 10 minutes and some of them go all the way until 25 minutes. So it depends on the intensity of the light, how close they are, but shouldn't be going for too long. So again, here is the place where you're going to want to write your hypothesis that I stated earlier for you. And then if-then statement, tell me which one is going to go faster, which one will have all the leaf discs rising and risen faster. Is it going to be the cup with the water or the cup with the water and the sodium bicarbonate? Afterwards, we're going to be graphing data, and I will show you that as we get to that part. So the next video you're going to watch is going to be the setup and the actual data collection for this first round. I'll have a second video for a data collection on a separate data set. Instead of just comparing water with baking soda, we're going to compare a different variable change. And then I'm going to show you the graphs that you're going to have to actually plot, and then you'll write a report.